Okay, thank you. Thank you very much. Thank you to the organizing committee for inviting me and CTLGH here just to share our experience on the protecting, preserving, and restoring African poultry biodiversity using the different technologies that we are discussing here. So, of course, I will focus my presentation on the first 4 points, but we should have in mind that for us to have these biotechnologies deployed on the ground, it's very important to be complying to the local, national, and international regulations on access and benefit sharing. So we'll just pause there at the end and see how we put the discussion in the next phase. Yeah, in Africa, generally, when we are talking about chicken, we will hear people saying local chicken or indigenous chicken, village chicken, and everything is globalized in that terminology, just local chicken. But we are forgetting that in different parts of Africa, different agroecological zones, we have a great diversity of chicken, sometimes locally adapted to their to their environment. And that's why in different countries, we may have what we are commonly calling local chickens having different names. Of course, populations are giving different names according to the performance, the traits, the phenotype they are observing, and also according to the value they are giving to these animals. And that's why in different countries, these are just few examples, based on local knowledge, they have different type of chicken. And from that, we need to know, and we observe that the need to feed, from the need to feed the growing populations in Africa in general, we need to rely on these short cycle animals. It can be chicken, it can be pig, or some other short life cycle animals existing. And we also notice that with the demands in animal food, also we have increased production based on limited number of genetic resources, limited number of breed, which is really growing on the continent. And with that introduction of exotic breed, we have decline in the genetic diversity on the continent, and some of the breed that we can see here are little disappearing. And in different agroecological zones, we have some genetic diversity or genetic resources that we really need to conserve because their potential may be used for future development, for the development of the future poultry industry, not only in Africa, but worldwide. So that's why it is very important to locate, to target some of those animals and what we consider very important and make sure that they are crop preserved. So when we consider that what I'm saying and talking about regarding the adaptability of chicken to agroecological zones, you can see some of the work done by our colleagues, led by Almas and Tadele and Olivier and others, how in Ethiopia, for example, it's possible to have this ecological niche modeling to identify where we have some animals locally adapted and for which traits particularly, and see how in different agroecological zones it should be possible to target some of the traits which are really important in local production system and see how we can sustain productivities in those areas. So the thing will not just be about improve breed wherever we want to send them, but you also need to think about appropriate breed for specific environment. So with this diversity and what we have, we recognize also that there are major traits that these genetic resources are undergoing. We have the rapid state of the homogeneous large-scale intensive production, as I mentioned. And most importantly, we have inappropriate management of what exists. Locally, very poor or no conservation program, and not appropriate policy to make sure that that genetic resources is well managed. And we also have, of course, from time to time outbreaks, like we have the Newcastle disease in local community, which is sometimes can be wiping out almost 85 to 100% of the population that exists. We also have the negative selections and the inappropriate breeding programs on the continent. By negative selections, wherever you are in Africa, when we are visiting a family in rural areas, to welcome you, most of the time they will be slaughtering maybe or some chicken or donating one to you to take away. And you will notice that most frequently they will be selecting their best cut, their best animals for consumptions or to donate to you to take away. It's still for consumption. And this is what we really call the negative selections, where all the best animals are removed from the breeding system. And also we have different types of disasters, emergencies occurring on the continent. And this is the reason why we need to make sure that we have good conservation program in place. And when we are talking about conservations, of course, we have different type of or approaches for conservations, in vivo or ex situ in vitro. And when it is in vivo, ILRI also and collaborators, we have the Tropical Poultry Genetic Solution Program, which has also been doing conservation to improve use in local communities using the different community-based breeding programs. And we also have, we'll be focusing for these presentations on the conservations using the primordial germ cells, which are presenting the advantages of giving us the potential to crop preserve large number of animals or of populations in very small place and make sure that, of course, we can protect them from different risks that I mentioned earlier. And we should be able to revive them from the biobank or for the biorepositories, wherever we want them, or to include them in the breeding programs as we want. So at ILRI, Nairobi, this is what we have been doing. Of course, building also on what our colleagues from Roslin Institute has been working on. So we isolate from different ecotypes of chicken in Africa, the primordial germ cells, either from the blastoderms for circulatory blood or from the gonad, and have them crop preserved. We have the backup at the country level, of course, but we have also part of it at the ILRI in our biorepository, and we re-inject them into any host. We are using the white leghorn as host currently, and we can recover them at whenever we want. Currently, what we are using, as I said, is the white leghorn. And when we know that when we are injecting the primordial germ cells, normally they are competing with the endogenous germ cells. That's why we are having the surrogate, the chimera chicken. But to recover using the chimera chicken, we need different generations of crossbreeding. So it is already good to be sure that we can have the chimera and crossbreed to come back to the indigenous chicken, but it will take time. And that's why when we have, I mentioned it, we have been working in different countries. We started piloting in Kenya, Ethiopia, Tanzania, but now we are fully working with the Kenya Agricultural and Livestock Research Organizations because this is a national program, they have their own breeding program, a national agenda, and they have different ecotypes of chicken in Kenya on stations, but they were not having any measure of protecting them in case of any risk, any catastrophe happening. But they also have different improved line that they have developed in station that they need to crop preserve. So this technology is coming to help them. But as I mentioned, with the chimeras, we will have different generations. We need different crossing to be able that we bring back the material that we have been crop preserving in the biorepository. But with what Mike Magro teams and at the Roslin Institute have been doing, developing the surrogate chicken and they have been working on the DDS4 and the ICA-space non-surrogate technologies. So from that, they are able to develop the surrogates that we should be using now at ILRI to be sure that in a very short time, we should be able to recover the chicken that we have been crop preserving. And the proof of concept, everything is well done at Roslyn, and we are transferring it to Nairobi. And of course, we collaborate with the National Biosafety Authority, as we are also collaborating with the Kenya Livestock Research Organization to make sure that whatever we are having in our biorepository, we can bring it back to the communities as they want. And our agenda is that populations, or the local communities are well connected or well attached to their genetic resources. They are the custodians, they are the ones who have been keeping them for generations for their own reasons. If we succeed returning to them what is considered as their own material, for them this has no value. It is very important for them to make sure that, yes, they have, or science is helping them to recover their populations, to assist them in meeting their needs and animal source food, etc. And this is what we have been doing. And at Cairo level, for example, I mentioned that they have their breeding programs to disseminate to different regions and neighboring countries what they have as local chicken. These are local lines developed on their stations. But now the demand for these animals is so high that for counties to order, they have to pay and wait sometimes 3 months before they are supplied. So it is very important to know that the demand is high within Kenya already, but Rwanda, Uganda, Tanzania, and many other countries are requesting for the material. While using the surrogate technology now, it is very possible to use the body machinery of the surrogate, because we need to compare that. Local chicken, they produce a maximum of 90 to 100 eggs per year, for example, in the best case. But with the professional layers, how many eggs can they produce? How many chicks can they produce per year? So we use that mechanism to make sure that applying this model of tropical poultry genetic solutions, we can have large-scale disseminations of the preferred breed by the community and return to them exactly what they want to meet their need in the food security and poverty alleviations, etc. Of course, I mentioned the fact that this material that we are biobank crop preserving is not just stocking them and returning. We need also to use that to support all the different technologies that we are mentioning here. If Hendricks or whoever want to come to ILRI, for example, and use some of the material for developments, or they have identified some of the traits that need to be developed, etc. How do we collaborate? How do we make sure that the cells are available to them and collaborate to explore the genetic potential of the genetic material biobank of the local animal genetic resources, and really use it to develop whatever we want here. It can be used for gene editing, it can be used for different technologies, etc. Of course, that platform is not only supporting biobanking for chickens, it is a biorepository that was created for different other research at ILRI, including One Health. And during the COVID, just to mention it here, that the platform has really been helping the local government in Kenya to identify some of the circulating strains of the SARS-CoV in Kenya, and of course, supporting research in that line. It means what we can be doing here can be applied also with what we are doing on chicken can be applied to other livestock. We are starting a program also on pigs. I know John has been working with our colleagues on also planning to work on cattle and some other livestock resources. So and even on wildlife. So it is very important to have it in mind. But as I said, transfer of material is very important. Not only genetic material itself, but the digital sequence that is coming with it, and sometimes the traditional knowledge going with that material. That's why we need to have in mind the context of Nagoya Protocol, access and benefit sharing in Africa in general, where we are supposed to work, and in Kenya in particular, and see exactly what is needed if we want to move the material we are keeping in Kenya to any partner elsewhere. If a partner wants to bring some material to Kenya for work, we need to have it in mind. So I'm not closing the conversation here. I'm just pausing so that later on we can be discussing about the access and benefit sharing regarding the movements of this material. Because if you really want the biotechnologies that we are working on and the genetic materials available to be serving globally, we need to be thinking about this. Thank you very much.